leukocidins. What do? How they work? They what? They kill leukocytes. And which leukocytes are they really going to kill? The phagocytes, right? Because how do they kill it? Yeah, good. They make the lysosomes to leak out into the cytoplasm and degrade the cells. Any questions about that?
Wait, can you say that again?
Yeah, so leukocidins, they are excellent enzymes that are secreted by bacteria that have been phagocytosed. And so if I'm a bacteria, I've been phagocytosed, this room is the phagosome, the phagolysosome, let's go there. And all those enzymes are coming in to eat me up, but what I do is I have enzymes that poke holes in this roof. And so that means all the digestive enzymes will leak out through the hole in the roof and actually kill the cell out there. And then I'm released. So, you know, it might not just be that I'm trying to kill the leukocyte. What I'm trying to do is just get out of the phagolysosome. So I'm poking holes in it so that I can get out. When I do that, all the digestive enzymes leak out as well. What are some other ways to evade the host? Cell wall. Give me some examples about cell wall. Yeah, that's more like the capsule. Cell wall. Oh, the M protein. What's the M protein? What does the M protein do? Okay, binds to H factor. Where's the H factor? Where does it get the H factor from? Our cells. If you got a bacteria that has M protein, it's going to steal H factor from our cells, and why would it want to do that? So that it inhibits or stops C3b. Okay, without C3b, your complement system doesn't work, right? Okay, so we're talking about evading the host immune system. How does trypanosoma do? Antigenic drift. Good. Like high five you, man. Because I told you, right? Remember trypanosoma. What diseases trypanosoma cause? Sleeping sickness. Yeah. And the way that it stays in a person's body and doesn't let the immune system clear it out is because the antigens on its surface are constantly changing. So you go, it takes you a week to 10 days to make antibodies or to make T-cell receptors and stuff, or CTL cells. And so within that time, and just within a week to 2 weeks, the antigens will change. Every week to 2 weeks. That's pretty cool, except for if you're sick, right? Okay.
How many questions are on the test?
Not that I have it up here. 50 questions. It's shorter than the other ones. Yeah. Which is the sorry, it's not reading myself. What the heck? Which of the following is
Why'd you stop?
which of the following is the because I couldn't remember what it said. Which of the following is the most common bacteria found in nosocomial infections? Which is the most common bacteria, not not Staphylococcus aureus, because that will be on there. Staphylococcus aureus is coagulase positive, but it's coagulase negative Staphylococcus. Is that okay? Everybody got that? Because I don't know if so let me just say it again. So the bacteria that's found most often in nosocomial infections, or the one that causes the most nosocomial infections, is coagulase negative Staphylococcus. Staphylococcus aureus is coagulase positive, so it's not one of those. Okay? Yeah, I'll repeat it a couple more times because I want you to get this, okay? So the bacteria that's most often causes nosocomial infections is coagulase negative Staphylococcus.
Is that like a lot of your UTIs and stuff?
Yeah, UTIs, but you know what really it is? It's, we're putting a lot of hardware in people now. You know, screws, pacemakers, and it's those type of bacteria that really bind to those kind of things and make this bacteria film that's really, really tough to just, it's really hard to kill with antibiotics, that bacteria film. What? It's not an endospore, it's just this slimy, it produces this really thick glycocalyx that inhibits stuff. Okay, good. Now, what is the, let's go to the next obvious question. What is the most common nosocomial infection in the body? UTIs. UTI stands for? Just in case I have urinary tract infection and we're like, where's the UTI? Okay. What's the etiological agent for septic shock? What is the etiological agent? What?
Lipid A.
Yeah, yeah, good. You're right, it's lipid A, but let me go back and ask this, what does etiological mean? The cause. Yeah, so instead of, in medical terminology, instead of just asking, what's the causative agent? That's how you and I would talk, maybe? And, but they would say, what's the etiological agent? Yeah, let me just write this on the board.
Oh.
Etiological agent, what's the causative agent? Okay. And the question was, what's that etiological agent for septic shock? And someone said, lipid A, right? Okay, is contaminated water considered a fomite?
No.
It's not, but water's not living, but it's not.
It has to be like a
Inanimate object, yeah. Okay, so, yeah. It's a what? Yeah, it's a, be careful because we don't want to, you're right, but be careful of our terminology because when we say vehicle transmission, we don't really need a fomite to do that. In fact, what kind of transmission is fomite involved with? Indirect. Okay. What about pus? Is that fomite? What about the pin that's in your hand? Is that a fomite?
Yes.
Yeah. So, you know, mostly it's just man-made stuff that we think of. No, I guess a rock or something could be a fomite. I guess. So, okay, which, there's two different types of toxins, right? What that bacteria produce. What are the two different types of toxins that bacteria produce? Endotoxins and exotoxins. An AB toxin, is that an endotoxin or an exotoxin? Okay, it's an exotoxin, good. And how does an AB toxin kill cells? What's the mechanism?
B touches a hole, A goes in, right?
Cool, yeah, great. So B binds, so AB means it's two polypeptides. An A polypeptide and a B polypeptide. The B binds to the cell, so it's the specific part, and the A is thrust inside and kills the ADP. Yeah, good. It kills the NAD, creating ADP ribosyl group? Yeah, so really what you want to know for the exam is ADP ribosyl group. How does an AB toxin work? ADP ribosylation. Do fomites make good diseases? No, I'm just kidding. Do fomites make good reservoirs?
So,
Do fomites make good reservoirs?
not really.
No, fomites don't make good reservoirs. Fomites aren't reservoirs. Okay? Question number 16. This is an old exam. It's not going to be question number 16. It could be. I don't know. Probably not. It wasn't the exact question. Kinase. What is kinase? Breaks apart clotting factors. In mechanisms of pathogenicity, where would it fit in? Which of the 4 mechanisms of pathogenicity? Exoenzymes, and exoenzymes would be evading the host. Okay, so let's do it the other way. Evading the host, exoenzymes. What was it? Kinase, thanks.
That's a recommendation. Can we answer these questions a little more assertively? Guys, do you mind? That's all I hear. Blah, 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 blah. I want to remember that on the test. Just a request, that's all.
Speak thoroughly.
Just listen.
And then make sure Adam got it.
Clearly. We'll get some crazy looks, but makes sense.
Okay, so so we got evade the host, exoenzymes, we got coagulase, and coagulase is going to cause a fibrin clot to form around the bacteria so it shields itself from the immune system. Okay. What's normal flora?
That's a nice area that's fine and it would, you know, new area.
Adam, what do you have to say about that?
It might take some practice.
Raise our hands.
What?
Let's raise our hands.
Let's raise our hands. Let's raise our hands. Okay, so yeah, so normal flora is bacteria that are normally found on or in your person. Right? Normal. Another name for that is normal microbiota. What about flora that or bacteria that you find just sometimes on you? Yeah, what I'm after there is transient flora. Transient flora is normal flora that comes and goes. Or it comes and then goes. Yeah. Right? So depending on the season, you can have different bacteria flora on you. Okay. Commensalism. Commensalism. A type of what? Type of symbiotic relationship. So when we're talking about bacterial infections, right? If the bacterium, like let's say for example, Streptococcus pyogenes on your throat, causing strep throat, what kind of symbiotic relationship is that? That's parasitic. So give me an example of commensalism. Yeah.
Little shark sucker thing.
Shark sucker
cleans
thing.
it off. Never mind. So it does purple.
Give me an example that's in your body, if you can think of one. Yeah, so commensalism, give me an example of commensalism, that type of symbiotic relationship in your body. E. coli, is that commensal?
Well, it helps break down stuff.
What's the difference between commensal and mutual?
Mutuals have both the benefit, commensal is
Mutual
one
and both
benefit.
benefit. So would E. coli be mutualism or commensalism?
Mutual.
Mutuals.
It's mutualism, right? I mean, the E. coli get in a nice warm place, lots of food come down the tube. And then they help us by breaking down vitamins and allowing us to digest vitamins and stuff. Yeah.
Are there bacteria that we know of that do not affect humans? Like they'll benefit from us, but we don't have anything to do with them at all?
Yeah, you know, here's one for you, okay? So there's one called Corynebacterium. Can't remember his last one, but there's a Corynebacterium that, this ring a bell? Where is it found? Because I've talked about it just a little bit before. It's found in your eye, right? What's it doing in your eye? Yeah, it's just eating. Yeah, it's eating the sloughed off cells from your eye. Lots of us actually have little mites that's in our eyelashes that just feed off the dead sloughed off, you know, because our stratum corneum is constantly sloughing off, and they're eating off those dead cells as well. So, yeah, we're just like rubbing them off now. And so we don't even know they're there. Yeah, so, the worst things come on my mind. You ever did those butterfly kisses? Yeah, no more, huh? Spreading those little mites. Those are examples of commensalism. Commensalism is just organisms that are helped by being on you, but you don't even know they're there. They don't hurt you or they don't help you. That's commensalism. You know, where one organism is helped and the other organisms is neither hurt nor helped.
So normal flora would be because it prevents us from getting sick and
Yeah, we always say that, right? That the normal flora is, you know, helps us from not getting sick and stuff. But actually, normal flora is any bacteria that's normally found on you. So these Corynebacterium that are in your eye, they be considered normal flora as well. So normal flora are generally, you know, mutualistic, but some of them are commensal. And if they get in the wrong spot, then they become parasitic. What's more powerful, endotoxin or exotoxin?
exotoxin.
Yeah, meaning that it takes less exotoxin to kill you than it does endotoxin. Okay? What's is it easier to? I don't know how to say this question. Which one's heat liable? Exotoxin means they're heat sensitive, right? Exotoxins, you can heat them up. Why? Why are they so sensitive? What's about? Why are they more sensitive than endotoxins? Because they're proteins, right? Proteins denature in heat, and endotoxins are lipids. And lipid what? Lipid A. Good. Where are endotoxins found? I mean, where's the lipid? Yeah. What type of bacteria produce endotoxins? What type of bacteria produce exotoxins?
Both gram-negative and
Gram-negative and
gram-positive.
gram-positive. Yeah.
Can we go over DNA and RNA viruses?
Yeah. Yep. So, I will ask you, what is the DNA virus that we learned? What's its name? That virus, right? Is that what you said?
Yeah.
I don't know that you articulated that clearly. That virus is a DNA virus. Good. So if this is a papillomavirus, what type of cell do you think this is? This is an epithelial cell, right? I just said that, I don't know, I'm just trying to make you guys think. That's not on the exam. Don't be like, thinking, oh, I didn't know that. Just, I did it. Don't worry about it. But anyway, that's an epithelial cell. That's a human cell, right? And then tell me what to write. Okay, so we got a papillomavirus. What's the first step? Attachment. Okay. So I'm going to attach it like this, right? Enter through portal of entry. And, wow, this is good. How's a papillovirus, what's a papillovirus's portal of entry? Yeah, it's either skin or mucous membrane, depending on where you're getting it, right? Yeah, so if papilloviruses cause warts. So if it's just warts on your hand and stuff, then it's going to be portal of entry through the skin. Okay, so portal of entry, wart cell entry. Okay, now it's in. Now it's going to bind to this epithelial cell. That's the attachment. That's the first step. What's the second step? Penetration. And is it just the DNA that's going to penetrate, or what? Anybody?
Yes.
Just the DNA? What's the third step? Let's just skip that question for a second. What's the third step? What? You're skipping a whole bunch of steps. A whole bunch of steps in between. So you can, what? Okay. Obviously.
Uncoding the DNA.
What? Uncoding! Did you find that in your notes, or you just like...
I just remember.
Okay, so if it's... The third step is encoding. Let me ask that previous question. Does the whole thing come in, or just the DNA? Yeah, the whole thing comes in, right? And so on animal viruses, the whole thing gets in. It's endocytosed by the cell. And that's the penetration part. Bacteriophages are totally different. They just squirt their DNA into the cell. Their coat stays out. So there is no encoding. On the exam, I ask you, which of the following steps do animal viruses have, but bacteriophages don't have? And the step that I'm talking about is the encoding step. Does that make sense? Because in bacteriophages, the coat stays on the outside, and so it doesn't need to encode. Okay, so uncoding. So uncoding just means that this capsid falls apart, and this is DNA, right? All right, good. Now, now what? Okay, early transcription and translation. So, which means what? Early transcription and translation. Tell me what's being transcribed, translated. What?
Your DNA is copied.
Good. Now, but DNA is not transcribed or translated, right? So what's, you're talking about transcription and translation, that's protein stuff. So you're on the right track. Enzymes to make more DNA. Good. So early transcription and translation to make the enzymes to copy the DNA. Does that make sense? So early transcription translation makes, or to make, the enzymes to copy the DNA. So what we want is a bunch of this. Okay. What's the fifth step? Late transcription. / translation. And this transcription translation, what are we making? For proteins for the capsid or for the coat, right? Because remember, I mean, with viruses, there's really kind of a lot of viruses are only made up of two things: DNA and capsid. So on this one, which is made up of just a DNA and capsid, first thing you're going to make is the proteins to make more DNA. Then the second thing is the capsid part. Okay. Steps 4 and 5 together, what can we call that? 4 and 5 together is biosynthesis.
They're just going to the ribosome and they're making these proteins.
Yeah, ribosomes. Ribosomes are always red. Yeah, and so this DNA, this DNA has to be transcribed into messenger RNA, right? That's where that's going. So the messenger RNA comes up here in the ribosomes to make the enzymes, the proteins, so that you can copy this DNA into these guys. And then the same thing happens with the legs. Yeah.
So on the test, did you ask us like what step comes next, or like how would you ask us questions like these steps?
I could ask that. I didn't. I'm like, hey, that's good. I could have had 51 questions. More is just like, well, in a way, I mean, what happens between the encoding and what's after 5? The maturation, right? So I'll put 6. I do ask you what's going on in early transcription, translation. And I can ask you what's going on with late transcription translation. So maturation is step number 6. And what's happening in maturation? You're just assembling it all together, right? I can't think of how to spell assemble, something like that. So assemble the viroids together, meaning put the capsomers, the proteins that make up the capsid, and the DNA together. And then 7 is these.
So 6 maturations are done in the ER.
6 maturation, this happens in the cytoplasm. Because you got all the proteins and stuff. I see what you're saying. Yeah, but all the proteins and stuff may be made in the ER, but then they're excreted out and they assemble it together on the cytoplasm.
Which cycle is this again?
So this is the cycle. This is the yeah.
Just DNA synthesis.
Yeah, so that's a really good question. Because everybody gets like, this is all coming like, so lysogeny and lytic cycle, they're only for bacteriophages. Bacteriophages, just to make sure everybody understands, those are viruses that attack bacteria. So when we talk about lysogeny and the lytic cycle, that's just for bacteriophages. Though there's a lot of things that happen in bacteriophages that are similar to the animal viruses. That's why we talk about it. And then also we can use in science, we can start using bacteriophages to infect bacteria, to make bacteria do what we want, either to kill them or to produce something that we want them to produce. Let's say we want penicillin, you want some type of bacteria to produce penicillin. You know, stick a penicillin gene not into an animal virus. You have to stick it into a bacteriophage. You have to pick a bacteriophage that's going to go through like sod. All right. Now this is the RNA virus that we talked about is what? What is its name? Yeah, for coronavirus. Hypo means small. So literally a small RNA virus. And it causes what disease? Polio. Okay. So polio can bind to either your epithelial cells of your mucous membrane, your intestinal tract, or your neural cells. Okay. So here's the by coronavirus, and what's inside it? Positive RNA strand. Okay. So we just go like that. I'll go positive RNA, and oftentimes you'll see SS, positive RNA strand. SS stands for single-stranded. Because believe it or not, in some viruses you can get double-stranded RNA. Okay, so single-stranded positive RNA, and you're going to just to kind of go through this a little bit more quickly. When you get attachment, you get penetration. Then you get encoding. And now what? Sorry, say it again.
We go into the ribosome and make a negative strand of RNA.
Okay, so we're going to go into the ribosome, and we're going to.
Create an enzyme that will create.
Yeah. Okay, let's do that then. So we're going to take this. We're going to go take it to the ribosome. And this is your positive stranded RNA. And it's going to come out the positive stranded RNA. But what is it going to make in the ribosome? An enzyme. Good. This is my rendition of a viral enzyme. And that viral enzyme is good for transcribing RNA. Viral RNA polymerase. Okay, so now what this is going to do, this viral enzyme is going to do what? It's going to start transcribing this thing, so it means that purple here. It's going to make a negative strand RNA. Yeah, that's when we got everybody up trying to do it down. I'm going to make that so you can actually read it. Negative RNA. Okay. And so that's just the, right, that's the complement to the positive strand. The positive strand RNA just means that it can run through the ribosome and actually make a protein. This negative RNA can't do it, so why are we making negative RNA? Good, because once we make a bunch of good negative RNAs, they become templates to make positive RNAs. Over and over and over and over again. So we just start filling up our cell with positive RNA because we're using these templates to make them. Remember, that's the first thing a virus wants to do is replicate its nucleic acid. Okay? What's the second thing the virus needs to do after it replicates its nucleic acid? Make its coat, right? I mean, remember, virus is just made of nucleic acid and protein coat, or capsomer. And so now, you got to make the capsomer. How's it going to make the capsomer? Using the positive RNA strands. So you just take these guys, start running them through all your ribosomes, and your ribosomes will read that positive RNA strand and make all of these capsomeres. No, that's a square. Anyway, it's going to make these guys. Okay. And so, I guess I stopped numbering these. So, 3, taking our positive RNA, going and taking it through the ribosome, we'll call that step number 4 here. And then RNA to make your DNA, that's step number, RNA, positive RNA to make your negative RNA, that's step 5. Making your negative RNA to positive RNA, that's step 6. This is not an exam, I'm just trying to help you see it, how it goes. Questions? Because I just ran away with it. Because what?
What's one of your cells when it catches this isn't happening
Yeah, this is happening in the cytoplasm. And so, one of the, what I'm doing here is just concentrating on making your positive RNA. And making the viral enzymes to just transcribe the positive RNA to the negative RNA. There's a number of genes that are turned on here, and that are red here, and one of them is another enzyme that inhibits those nucleases that are in your cell. Okay, so the big thing that is here is just remember what that picornavirus is a single-stranded positive RNA. Therefore, in order to replicate itself, it has to transcribe a negative RNA to make it as a template. What am I talking about? So you've got the single-stranded positive RNA. What you need to remember is that you're going to make a negative stranded RNA as a template to make a bunch of positive RNAs. That's the most important thing to remember about this on the exam. Yeah.
I'm still a little bit confused with the DNA thing. So it comes in to the cell and it has its DNA. Does it go to the ribosome? Does it go to the nucleus? And then it makes an how does that work?
Yeah, so this DNA, it goes into this part right here is in the nucleus. So when you're copying DNA, that's always in the nucleus.
And is it using its own enzyme or the host enzyme to make an mRNA strand?
Host enzymes here, not its own. Okay, someone with measles. Is there any more questions on this first?
Yeah, so we need to know specifically that this is the coronavirus and this is the...
Yeah, do. Okay, yeah, know specifically this coronavirus and that it gives polio. And this is the poliovirus, it makes warts. Measles, what's the mode of transmission for measles? Let me describe it for you, okay? So measles can stay airborne for 2 hours and float all around the room. So what's the droplet is great answer because that's because it's wrong. It's really lots of people will say that. So why is that wrong? Yeah, drop it just a meter. You own it until it's a meter away from you. You drink out of someone's water bottle and you get mono. What's the mode of transmission? Indirect, because what's the phone line? Water bottle. Okay. I think you guys will all, whatever example I use, you will all get direct contact really easy, right? So if I say kissing, handshaking, that's direct contact, right? A harder one might be a mosquito bite. What's that? Vector biological. What about if flies are in your soup? Vector mechanical. Okay.
Sorry, what's the difference between those two?
Vector biological is when an insect bites you, and actually the portal of entry would be the parenteral route if it's going to be biological. Because mechanical, it's going to probably be mucous membrane because you're going to eat it, and then it's going to get into the mucous membrane. My daughter wanted to make homemade ice cream yesterday. She found the little ice cream maker. It was out in the garage, and there was like a dead mouse. So that would be if they made ice cream and they ate it, and they would be vector mechanical. Direct phone. In fact, I was supposed to bring that canister. I was going to put it in the autoclave. It was even a little girl serving that, but I'm not going to say it. Please share. She's like, Dad, there were 3 mice in there. And I took it out, and the first one fell out. The second one I had like go like that, and then finally it fell out. The third one I had to get a stick to get it out. So like, don't even touch that. I'm going to take it to school and autoclave it. And then sell it. Okay. If someone is talking like up here and they spit and it lands on you in your eye and you get an eye infection. And that is the question, how far did you spit, right? So if I'm back here and I hit you, it's, hey, after it here, I'm not responsible for it. So it's airborne, man. Oh yeah, so if it's airborne, if it gets more than a meter, then it's airborne. If you sneeze right in somebody's face, then it's, and when you have kids, that will happen. And that will be droplet, right? Viruses are grouped into classifications how?
I had a question for you, Leslie. If someone sneezed into their hands, and you shook hands with them, would that be direct contact?
What do you guys think?
Direct.
If it's indirect, it has to be a phone line, so it's not indirect.
I think it's direct.
It's direct. Yeah, it's direct.
It's direct.
It's no longer droplet, it's on your hand, so it's direct. So what did I just ask? Oh yeah. How do you classify viruses?
Shape.
Nucleic acids, shape. The shape is the, what's another word for shape? Morphology.
Yeah.
And what's the third one? Yeah, whether it has an envelope or not. Okay.
Say those again, please.
Yeah, so it's nucleic acid type, nucleic acid, morphology, and whether it has an envelope.
What are we going to know about
Koch's postulates. Koch's postulates just know the order in which they come in each step, okay? And also know when Koch's postulates does not work. So Koch's postulates doesn't work if there's more than one organism that's causing the disease. Because Koch's postulates, it's based off getting a pure culture off a sick or dead animal. And then putting that pure culture back into a healthy animal to see if it makes it sick. So if you can't, if it's just one, if there's more than one bacteria, it's really hard to do that. What are some other reasons why Koch's postulates wouldn't work? Yeah, so you can't grow it up into pure culture. If there's two or more bacteria, or if you can't grow it, I just said that. Yeah, so if you can't grow it on media, you can't get a pure culture.
So that's related to smallpox with Dr. Would that be an example?
Yeah, the neighbor kid being the animal, right? Sort of. I mean, he didn't really do it because what would have really worked is if he would have took smallpox from somebody and without giving the kid a vaccine, then give it to the kid, and the kid died, and then that would be more possible. Because you have to repeat the disease. Yeah, you have to. Right, you have to take the organism out of somebody that's sick or something that's sick and repeat the disease with just that pure culture. That tells you that.
So this is narrowed down to
Yeah. Why are encapsulated bacteria so hard to for your immune system to kill? I'll say here's mumbling.
It takes piece of membrane, so it's encapsulated with this piece of this.
That's an envelope.
Wait, what did we ask?
What was the question?
What's a capsule? Why are encapsulated bacteria so hard to the immune system to die? Right, because the capsule covers up the antigenic determinants of the bacteria. So your antibodies, for example, can't attach them. This is one, I got one that nobody knows here. You got something, Chris?
What's the process of where a virus picks up an envelope?
Yeah, that's in the release. Some viruses will take the membrane with it, that's all.
There's no term for that?
Funny.
Body? Oh, it is.
Yeah,
Body. That's not-
but they all bud. I mean, but some will, the membrane will stick to the capsid and some don't.
Oh, so there can be like without.
Yeah, yeah. It just depends on whether the capsid will stick to the membrane as it goes out.
So then budding is just the blowing off the shell of the capsule?
Yeah, so, well, budding is just it breaking out, and as it breaks out, it'll bud out as it does. But if the protein coat is unable to stick to that membrane, the membrane will just slide off it.
Okay.
And it'll go free.
So as soon as you say a virus picks up some of the membrane, and then that gets ripped off the virus dies.
Well, the virus is never alive.
Well,
The virus,
you know,
yeah, the virus
becomes
will,
innate.
yeah, the virus will be picked up by your immune system a lot easier. I mean, you just gotta remember these are chemicals that are sticking to your cells and just replicating inside them. So it won't be as effective for sure. Because there's no receptors on the envelope itself to, oh no, I take that back. It'll become infected. It'll become innate. Whatever you said, yes. It will die.
It'll
Okay,
die.
we'll just say that.
Okay.
Okay, what was the question I was gonna ask? Oh, that nobody knows.
Sorry.
If you get this question, you don't even have to take your exam.
Yes, we do.
I'm kidding. You're kidding. Everybody's like, because there'll be about 10 people. I think that there will only be about 10 people in the invasives. What are they? Invasives. Okay, let's just dump the ante here. Anything I guess it doesn't. No.
Got it.
Streptococcus myces,
Okay. And what-
mutagens,
What?
and mutagens,
And what about a,
they form cavities.
no, it's something else.
They're like bacteria,
Okay, you gotta articulate?
Oh.
I know Adam's right there, but,
Oh, enunciate, bacteria.
oh, enunciate, yeah.
Proteins that promote the ingestion of bacteria by non-pathogenic cells.
Awesome.
Take the test.
Automatic edge. Yeah, okay, so what that is you have bacteria that have these invasive proteins. And the invasive proteins bind to normal cells of the body and get those normal cells to phagocytose them.
Phagocytose themselves?
Yeah, so like if this is a normal cell body, normal cell, like an epithelial cell right here, and I am a bacteria, this is not gonna work so well, but I have these invasins on me, and when they stick here, it causes this cell to think it's a phagocyte. And so it swallows me up inside. But the thing is with normal cells, they don't have really good phagosomes. And so I can break out of their phagosome and start living inside them. Yeah, those are invasive.
Are they on viruses or bacteria?
Invasins are in bacteria.
So what should we look for in the end on the test, like for how that's written? Like just like non-pathogenic cells?
How about something like promote the ingestion of bacteria by non-phagocytic cells, something like that.
Is there a number or a letter?
There it goes. That means that now there's going to be a lot of you get 100%, because that was the one that killed the 100%ers.
What question?
On this exam, it's question number 41.
What's the letter?
All right, let's see if there's anything else. What is an example of microbial antagonism? What's microbial antagonism? Yeah, that's perfect. Like normal flora can produce chemicals and stuff that's kill pathogens. They're called bacteriocins. Bacteriocins are chemicals that kill other bacteria. Let me say that again. Bacteriocins are chemicals produced by bacteria to kill other bacteria. It might be spelled wrong.
Oh, okay.
That's an example of microbial antagonism. Okay, so make sure that you, we're out of time, but make sure that you got hard, stale, and DIC, disseminated neurovascular coagulation. That's all part of septic shock. Okay?